Alrighty, this is the micro final part 3, I believe, or 2, not quite sure, maybe 2. Structure of the DNA. There are 2 strands. The 2 strands are strings of nucleotides. There are hydrogen bonds between the 2 strands. The neighboring deoxyribose is connected. 3 of one deoxyribose to 5 of next deoxyribose. That's the position on the strands. They are a double helix. When you're talking about anti-parallelism, this is the way the sugars and the phosphates come together on the backbone. One side of the helix runs in the opposite direction of the other. The order of the bond between the carbon on deoxyribose and the phosphates is used to keep track of the direction of the 2 sides of the helix. So one helix runs from the 5 to 3 direction, the other runs from 3 to 5. DNA runs from 3 to 5, and the complementary side is 5 to 3. Remember the base pairing at the golf course. A goes with T, G goes with C. Lipids. Triglycerides, phospholipids, and steroids. A triglyceride is an important storage lipid. Example would be fats and oils. A glycerol backbone plus 3 fatty acids. That's a hydrocarbon chain. It's bound by an ester bond. Phospholipids serve as a major structural component of cell membranes. Remember the phosphate head and the glycerol neck plus the 2 fatty acid tails? The head is polar and hydrophilic and therefore loves water. The tail is nonpolar and hydrophobic and hates water. Phospholipids in a single layer, it forms a circle. Bilayer, heads on the outside, tails on the inside. Basic DNA replication. You need an accurate copy so that information can be passed on. Only a small region of the DNA is separated at a time to preserve the original parent DNA molecule. This process is called semi-conservative replication. The old parent strand is the template. The new daughter strand is the complementary plate. The replication fork contains multiple enzymes that form the replication apparatus. The replication fork has 3 functions. 2 strands of the double helix are unwound. I should say the 2 strands of the double helix are unwound and separated by the enzyme helicase. So the helicase separates the 2 strands of the double helix, which disrupts the hydrogen bonds and then exposes the nitrogen bases to base codes of DNA. DNA polymerase 3 proceeds along the DNA molecule. It can only lengthen the strand in one direction, 5 to 3, attaching the correct nucleotides according to the pattern of the template, A to T and C to G, releasing pyrophosphate. The resultant new DNA molecule contains 1 strand of the newly synthesized DNA and the original template strand. The DNA helicase is what moves the replication fork. DNA helicase moves the replication fork. The replication fork needs 3 additional enzymes in the 3 to 5 direction to lay down RNA. Primase, which is synthesizing an RNA primer. DNA polymerase 1. It destroys the previous RNA primer while replacing it with DNA, closes the gaps and repairs mismatches, and DNA ligase seals the gaps between the fragments. So the replication fork needs 3 additional enzymes in the 3 to 5 direction to lay down that RNA. And in the 3 to 5 direction, it's primase, which is synthesizing an RNA primer, DNA polymerase 1, that destroys the previous primer, the RNA primer, while replacing it with DNA. And that DNA polymerase 1 closes gaps and repairs mismatches. and DNA ligase seals the gaps between the fragments. 3 types of RNA: mRNA, tRNA, and rRNA. mRNA is a transcript or a copy of the DNA, the template which later is read as a series of triplets called codons. So the mRNA is a transcript or a copy of the DNA, template which later is read as a series of triplets called codons, composed of 3 nucleotides. tRNA transfer amino acids used to build the protein translator. It binds the mRNA and amino acid, determining where the amino acid will be placed in a protein. Basically, tRNA is also a copy of the specific region of DNA, but it folds back on itself so it looks like a clover leaf. It's an adapter that converts RNA language into a protein language. The bottom of the clover leaf exposes a triplet, an anticodon, which will match up with the mRNA's codon. And then finally, rRNA, part of the ribosome, which is the site of protein synthesis. Barriers to protein synthesis. The cell membrane, drugs that can dissolve in phospholipids, which are hydrophobic, pass through, but most cannot and must pass through protein portion or use transport systems. CNS and the eye. Infections are hard to treat because of almost impermeable membranes and proteins that bind to drugs. They form a protein-drug complex that cannot pass membranes in this form. Binding is reversible. An unbound drug leaves the area, more is released from the complex. This slows down the distribution. Spontaneous generation was an early belief that some forms of life could arise from vital forces present in non-living or decomposing matter. An example of that would be flies from manure, etc., etc. Louis Pasteur showed that microbes caused fermentation and spoilage, and disproved spontaneous generation by use of the swan neck flasks in his experiment. Resolution. Speaking of the microscope, the ability to distinguish between 2 points measured as resolving power is dependent upon the size of the objective lens, the wavelength of the light, and the refractive index of the material between the objective lens and specimens. An example, oil has a refractive index of 1.5, and air has 1.0. So resolution is the ability to distinguish between 2 points measured as resolving power, dependent on the size of the objective lens, the wavelength of light, and the refractive index of the material that you're trying to pass it through. Beta-lactamase. Beta-lactamase breaks the bonds in the beta-lactam ring of penicillin, rendering it inactive. Penicillin is no longer able to prevent cell wall synthesis in the microorganism. So when you hear of penicillin-resistant microorganisms, it is because these little critters make beta-lactamase. Phases of growth. You have the lag phase, the exponential phase, the stationary phase, and the death phase. Lag phase is a relatively flat period on the graph when the population appears not to be growing. And this occurs primarily because the newly inoculated cells require a period of adjustment, enlargement, and synthesis. And the cells are not yet multiplying at their maximum rate. The exponential phase is when the cells reach the maximum of cell division, during which the curve on the graph steadily climbs. That's called that logarithmic growth, the log phase. Stationary phase is the population enters a survival mode in which cells stop growing or growing slowly. The curve levels off because the rate of cell inhibition or death balances out the rate of multiplication. Finally, the death phase, the cells begin to die at an exponential rate, literally perishing in their own wastes. The curve dips down, usually is slower than the growth phase. Nutritive growth requirements, CHUMPS. Let's talk about carbon. Carbon, and then a heterotroph is an organism that must obtain its carbon in an organic form from the bodies of other organisms. An autotroph is an organism that uses inorganic carbon dioxide as its carbon source because they have the capacity to convert CO2 into organic compounds. They are not nutritionally dependent on other living things. But a heterotroph, an organism that must obtain its carbon in an organic form from the bodies of other organisms. Sugars and polysaccharides can satisfy this need for carbon. Hydrogen, a major element in all organic and several inorganic compounds, including water salts and certain naturally occurring gases. Hydrogen maintains pH, forms hydrogen bonds between molecules, and serves as a source of free energy in the oxidation reduction reactions of respiration. Oxygen, obtained from various sources, organic and inorganic. Toxicity of oxygen, hydrogen peroxide, superoxide, and hydroxyl radicals. Nitrogen, remember these are the nutritive growth requirements. Nitrogen, main source of nitrogen gas is the Earth's atmosphere. To use the nitrogen, proteins must first be broken down to amino acids and nucleic acids into nucleotides. Phosphorus, the main inorganic source of phosphorus is phosphate, derived from phosphoric acid. Phosphate is a key component of nucleic acids. And sulfur, minor but it's essential. Sulfur is an essential component of some vitamins and amino acids. In addition to the CHOMPs, the nutritive growth requirements, growth factors in laboratory growth are added, amino acids and nucleotides, and trace elements of micronutrients like metal ions, zinc, and manganese. Measuring growth. The mass is the dry weight of the non-living culture. Turbidity is a simple observation that a tube of clear nutrient becomes cloudy or turbid as microbes grow in it. Measurement of turbidity, you would use a spectrophotometer. Measures absorbance of light by cells in broth culture. Total cell counts, counting chamber. It has a known depth and the squares of known dimensions. Count cells, specific squares determine number of cells per volume. And a viable stain distinguish live and dead cells. A viable cell count, platelet count, a plate count, or streak plate or pore plate. Culture is serially diluted. Dilution is placed in liquid agar. Bacteria grow and form colonies. You count the colonies. Accurate range 30 to 300 colonies per plate. Filtration count. Collect on a filter placed on plate, and count the colonies. Most probable number is a statistical method of counting through a series of dilutions. Quantitative, the polymerase chain reaction. Quantitative PCR, real-time PCR, used to quantitative viral load in blood samples. A probe has a fluorescent label. The more DNA you start with, template, the more fluorescent, therefore the more organisms is in the sample. Sterilization. Destroys all microbial plant life. The thermal death point is the lowest temperature required to kill all microbes in a sample in 10 minutes. Thermal death time. The shortest length of time required to kill all test microbes at a specified temperature. Moist heat in the autoclave, 15 psi's at 121 degrees centigrade for 15 minutes. That's steam under pressure. It kills all microbial life, including endospores. Dry heat requires longer periods of time for sterilization. 160 degrees for 2 hours, or this is Celsius now, or 171 for 1 hour. That was on the earlier test. 160 degrees centigrade for 2 hours, or 171 centigrade for 1 hour. Ionizing radiation, that's X-rays and gamma rays. Sterilizes by stripping away electrons. And that's used in food, drugs, vaccine, medical instruments, and surgical gloves. Disinfection, use of a physical process or a chemical agent to destroy vegetative pathogens, but not bacterial endospores, on inanimate objects, not for use on humans due to it being toxic. That would be disinfection, not for use on humans due to it being toxic. An example would be a 5% bleach solution to wipe down an examining table, boiling eating utensils used by a sick person, immersing thermometers in an iodine solution between uses. It reduces the number of pathogens. Moist heat and boiling kills most vegetative cells. Pasteurization is a technique in which heat is applied to liquids to kill potential agents of infection and spoilage, while at the same time retaining the liquid's flavor. All hail the holy milk in food value. Radiation, UV light, non-ionizing, that damages the DNA. Alrighty, lots of pages to go yet. An example of that could be germicidal lamps to cut down the concentration of airborne microbes in hospital rooms, school, food prep areas, blah, blah, blah. Ultraviolet irradiation of liquids is used to treat drinking water, purify other liquids. Decontamination renders contaminated surface safe to handle. Reduces the level of microorganisms so that the possibility of infection or spoilage is greatly decreased. Sanitation, any cleansing technique that mechanically removes microorganisms to reduce contamination to safe levels. Degermination, reduction of number of microbes on human skin. Degerming procedures are the surgical hand scrub, the application of alcohol wipes, the cleansing of a wound with germicidal soap and water. Antisepsis, kills the microorganisms on living tissue, are applied directly to exposed body surfaces. Example, preparing the skin before a surgical incision with iodine compounds, swabbing an open root canal with hydrogen peroxide, and ordinary hand washing with the germicidal soap. Leukocytes are white blood cells. They're involved in innate and adaptive immunity. Innate and adaptive immunity. And on that note, we're going to pause.